Hello, my name is Lauren Frederick. This presentation covers neural communication through neuron and synapse structure, function, and dysfunction. My research for this presentation came from the article The Cell Biology of Synapse Formation by Thomas Sudoff. Introduction. Neural communication is the process by which neurons transmit signals, allowing the brain and body to work together seamlessly. This communication underlies everything from basic reflexes to complex thoughts and emotions. It occurs through two main types of signaling, electrical and chemical. Electrical signals travel within neurons, while chemical signals transmit through neurotransmitters, allowing neurons to communicate across synapses. Sudoff emphasizes that neural communication is not just a simple transfer of information, it relies on highly organized molecular processes. Specifically, the formation and function of synapses, which are tightly regulated by specialized proteins like neurexins and neuroligands. These molecules guide the correct alignment of neurons, ensuring accurate signal transmission. Without this precise organization, neurons would not be able to communicate effectively, leading to disruptions in brain function. On the right here, we have a micrograph of a synapse. The red arrows are pointing to the neurotransmitters traveling from one neuron to the next. Throughout this presentation, we'll explore how neurons are structured, how they transmit signals, and how synapses play a critical role in neural communication. Here is the structure of a neuron. It is the basic unit of communication in the nervous system. Each neuron has three main components: the cell body or soma, the dendrites, and the axon. The cell body contains the nucleus and is responsible for maintaining the cell's functions. Dendrites act like antennas, receiving signals from other neurons, while the axon transmits electrical impulses away from the cell body toward other neurons or target tissues. The axon terminals are at the end of the axon, and they are what connect to the next neuron and create the synapse. This is where information is transmitted across a very small gap to the next neuron. Neurons form highly specific synaptic connections to ensure precise signal transmission. Synaptic specificity is essential for maintaining accurate and reliable neural networks. Sudoff highlights that this structure is essential for synapse formation. The axon is especially important because it carries signals to the synapse where communication between neurons occurs. Importantly, the ability of neurons to form specific synapses depends on molecular cues that guide axons to the correct targets. These signals ensure that the right neurons connect, supporting accurate and efficient neural communication. Synapse formation and structure. Neurons communicate using chemical or electrical signals, which are crucial for transmitting information across long distances. Chemical synapses use neurotransmitters to relay signals across a synaptic cleft. So here in our image, this is a synaptic cleft. These are vesicles, which will hold the neurotransmitters. They will move towards the end. They will open up and release the neurotransmitters to the next neuron. Electrical synapses directly transfer ions through gap junctions. At rest, a neuron maintains a resting potential. That is where the inside of the cell is negatively charged compared to the outside. When a neuron is stimulated, that is the initial contact, axons and dendrites identify and connect. An action potential is generated. That is when a rapid electrical impulse travels along the axon. This process involves the movement of ions, neurotransmitters, such as sodium and potassium, across the neuron's membranes. With synaptic differentiation, specialized proteins organize to form a functional synapse. When synapses reach maturation, they will stabilize and refine their function. According to Sudoff, this electrical signaling is closely linked to synapse formation. When an action potential reaches the axon terminal, it triggers the release of neurotransmitters into the synaptic cleft. This process depends on precise molecular mechanisms that align pre- and post-synaptic structures. So here's pre, this is post. Sudoff emphasizes that without these highly regulated interactions, electrical signals could not be effectively converted into chemical messages, disrupting neural communication. In this image, we can see the axon terminal coming to the next neuron. These proteins will have to line up in order to connect Once they connect, they become mature. The vesicles are allowing neurotransmitters to be released, and once they have released, it will separate again. Molecular mechanisms of synaptic transmission. The key steps in synaptic transmission start with vesicle docking. So again, these bubbles are called vesicles that hold the neurotransmitters. They will release across the synapse. Synaptic vesicles position themselves at the presynaptic membrane. When an action potential reaches the axon terminal, calcium influx triggers vesicle fusion, and it triggers the release of neurotransmitters across the synaptic cleft. Neurotransmitters bind to postsynaptic receptors, initiating a response. Then, neurotransmitters are removed by reuptake or enzymatic breakdown. Sudoff expands on this by explaining how synaptic adhesion molecules help anchor the pre- and postsynaptic sites. Without these molecules, neurotransmitters may not be accurately delivered to the receiving neuron. This article also emphasizes that synaptic communication is not static. Neuronal activity itself can influence how these molecules behave, meaning that the more a synapse is used, the stronger and more refined it can become. This dynamic process is crucial for learning and memory formation. Synaptic plasticity and regulation. Synaptic plasticity is the ability of synapses to change their strength over time in response to neural activity. This process is fundamental to learning and memory, and it allows the brain to adapt and store new information. Sudoff highlights that the synaptic plasticity is not just a random occurrence, it is a highly regulated process controlled by molecular and cellular mechanisms. There are two primary forms: long-term potentiation, LTP, and long-term depression, LTD. Long-term potentiation occurs when repeated stimulation strengthens the synapse, making future communication easier and more efficient. This mechanism is often associated with memory formation. In contrast, LTD, long-term depression, weakens the synaptic connections through reduced activity, which is important for forgetting unnecessary information and maintaining balance in neural circuits. So again, here we have a picture of LTP creating strengthening this connection, and LTD, which will weaken this connection. According to Sudoff, this regulation depends on activity, dependent signaling, and complex protein interactions. For example, molecules like neurexins and neuroligands play a key role in stabilizing and modifying synaptic connections. Without these precise regulatory systems, synaptic plasticity would become dysfunctional, contributing to neurological disorders such as Alzheimer's disease, autism spectrum disorders, and schizophrenia. Again, in long-term potentiation, we can see the glutamate was released, and since it's firing multiple times, it's going to react and is going to strengthen this connection. Here, it is going to remove one of the receptors, which is going to become less responsive to the neurotransmitter. Disorders linked to synaptic dysfunction. As we discussed, disruptions in synaptic function are linked to neurological disorders. Sudoff provides further evidence that dysfunction in molecules like neurexins, here in green, and neuroligands, here in purple, can contribute to conditions such as autism, schizophrenia, and Alzheimer's disease. If these adhesion molecules are impaired, the precise alignment between neurons is disrupted, leading to faulty signal transmission. So if these two cannot line up correctly, then the axon terminal cannot line up to the dendrite, so the neurotransmitters cannot be sent effectively. Understanding these mechanisms is crucial for developing targeted therapies for these disorders by addressing the underlying synaptic dysfunction. In summary, Sudoff's research gives us a deeper understanding of how synapses form and function. This process relies on specific molecular interactions and ensure accurate communication between neurons, showing the dynamic processes they must undergo. Furthermore, synaptic plasticity, how synapses strengthen or weaken based on activity, play a vital role in adapting to new information. The dysregulation of synaptic processes led to various diseases and even neuronal death. The insights from Sudoff enhance our understanding of neural communication and provide crucial context for exploring how synaptic dysfunction contributes to neurological diseases. This image here shows neurons over a 4-day period. We can see the initial layout of the network, and over the 4 days there are several lost connections. Those are shown in red here. There are also several new connections that were made as well, and those are displayed in green. If the neurons are not being used, then they will be lost and new connections can be made to transmit the information. Here we can see how dense the spine was, and over the 4 days the density did not change. However, the survival went down, so we did see connections that were lost here. We can see between day 0 and day 4, there were more that were lost than were gained. Here are the references that I used. The first 3 references were for the images, and the last one was the article that I used for my source. Thank you.